to introduce Greg Pintilie. And I'm really happy that he accepted our invitation to speak in our fall seminar series. Greg is a research scientist at Stanford University, working in the laboratory of Professor Hua Chu. And he's worked with Hua for about 10 years. The lab was previously at Baylor College of Medicine in Houston, Texas, but it moved to Stanford in 2017. Greg received his bachelor's degree at York University and a Master of Science degree at the University of Toronto. And he completed his PhD in electrical engineering and computer science at MIT in 2011. There, he was supervised by David Gossard and Jonathan King in studies that involved segmentation and molecular modeling of cryo-EM maps. And his research since has continued to focus on analysis of 3D maps and models from cryo-electron microscopy. And just one final, more personal note that we at Rutgers, of course, know him well because of his long involvement with the EM Data Resource Project, where he has made many important contributions, especially to our ongoing challenge activities. So thank you so much, Greg, for being here. I look forward to hearing your talk.
Thank you very much. So it is, thank you very much for that introduction. It's great to be able to present some of the research. And as Kathy said, a lot of it does come out of the EMDR project and the challenges we've been doing. So just a quick overview of what I'll talk about. So I'll just briefly introduce cryo-EM maps, what they are, how we do the reconstruction, what we mean when we talk about the resolution of a map. And then I'll get into this area that I've been looking at for a while, and that addresses this question is what is the resolvability of different atomic features, which is what we look for in these maps. So I'll go back a little bit to some of the work I did earlier in segmentation, because I think it's a little bit relevant. And then I'll get into this new Q-score analysis that we've started to apply, and we can apply it to atoms, side chains, ligands, ions, and water. So starting with cryo-EM maps, and before I dig in, let me reiterate what Stephen was saying, that if there are questions along the way, I'm happy to stop and make this more of a discussion. That would definitely help. I only have about 20 slides in an hour, so I'm gonna try to go slowly, and it'll be interesting to see what comes out. So cryo-EM, basically we start by imaging frozen particles, and we take images on micrographs. Now this is all sort of a simulated scenario, so usually the particles are not this well visible. But from the micrographs, we pick the particles and we try to arrange them into similar views, so the ones that look similar, and these we call class averages. And from the class averages, we try to find what is their projection orientation, so with respect to the 3D volume that we're looking for. We start by guessing this, and we refine these orientations, but eventually once we find the right orientations, we back project the 2D images into a 3D volume. So what do we mean by resolution? So the gold standard procedure for determining the resolution involves splitting up the particles into, splitting up a data set into half. So we take half of the particles or half of the micrographs, and from that we get map 1, and then from the other half we get map 2. So because this is an iterative process, we start with, even if we build a different initial model, that is ideal. So we want to keep the two data sets completely independent. And from half of the data set, we get one initial model, which we refine, right? So we add more particles, we keep refining the orientation angles until we see more and more details. So we might go down from 15 angstroms to 10 to 4 to 3, depending on how much detail are in the micrographs, and in this case, this is a map of apoferritin. So because this particle is so stable, and there's usually uh, um, there's a high degree of symmetry, so we can use symmetry as well to um, remove the noise. And basically we get down to something that might be 3 angstroms or 2 angstroms. And at 2 angstroms, we start to see things that look like the backbone and side chains. So these two maps look pretty much the same at a lot of these resolutions, even at let's say 2 angstroms of detail. They look pretty similar. However, there might be small differences in the small bumps and different features around them. But to calculate the resolution, what we do is we take the final maps and convert them into Fourier space. And in Fourier space, basically we can look at different we look at different frequencies basically, and the different frequencies or the different let's say the features at a lower frequency might look similar. So we do a cross correlation between these the Fourier converted maps, and at lower resolutions, if we do a correlation, we tend to see that they correlate quite well in this case. So that means that the 2 independent maps agree very well at these lower frequencies. And as we go to higher frequencies, it starts to drop. So they start to disagree. And this is where the effect of noise comes in. So because there's still a bit of noise in the noise in the micrographs will be different in the 2 data sets. So, and at those higher frequencies, that higher frequency noise will not correlate as well. So this FSC plot will start to drop. And there's this magical value that we use that is well established or commonly agreed upon of 0.143. And where the FSC plot goes below this value, at that spatial frequency or resolution was what we say is the resolution of the map. So that means that at this resolution, or these frequencies in particular, still agree reasonably well, let's say, between the 2 maps. So this is the commonly way of talking about map resolutions. Now, let me go back a little bit to how do we visualize these maps? And again, so we're looking in these maps, we're looking for atomic features, proteins, RNA, ligands, and things like that. But the first step, and this is something that I started to look at as I was getting into cryo-EM, is how do we segment the maps? And the process that I used goes back to this idea of watersheds. So if we think of a map, just an example, one-dimensional map, and here we can think of this curve as the height of the curve corresponding to the density, or let's say the value in the map. So denser parts of the map are higher, and then lower density regions are smaller. So so one way to break this map apart is to basically assign a region to each peak in this curve, and then the valleys, or the lowest point between these peaks is where we put the boundaries between them. So generally, because we do have noise in such maps, this can basically create a lot of different segments, which may or may not be signal. It could be noise. And so one way to deal with this is to look at something that is referred to as scale space filtering. So basically what we do is we take this one-dimensional curve and we smooth it. And the smooth curve is this red curve. And then we see for each peak, we do an uphill climb. And then we see which peak in this new curve does it fall upon. And then we group the regions based on the peak that it falls onto. And if we use Gaussian filtering, it's basically guaranteed that as you smooth it, you get fewer peaks. So that's kind of a good feature to have of this type of filtering. So we get a segmentation with fewer regions that maybe has grouped together either noise, or it could still be signal, but maybe higher resolution signal, as we'll see in a second. But what this does is now to look at an actual 3D cryo-EM map. So we have the map here on the left, and then this image here shows the watershed region. So this is these are the segments or the regions before any smoothing or any grouping has been applied. So you can see that there is a lot of little blobs, and each blob, much like in the one-dimensional case, you can think of each blob as corresponding to a peak, and then the boundaries between two blobs basically fall at the lowest point between those two peaks. And you can, and if you look closely, like some of the blobs might be attached to each other. So that means that they're actually in the map, that lowest value is such that they connect, or the blobs might actually not be, or on some sides, they might not actually be adjacent to anything. And this has to do with the threshold that is applied. So when we visualize the map, we have this notion of a threshold. So we only show the density values that are higher than that given threshold. Now, if we want to apply the smoothing and grouping, as I mentioned, we can start doing that. So after smoothing and grouping, now we get fewer regions, which are a little bit larger. And then in this case, this is a map of GROW-EL. And in this case, if we keep going with the smoothing and grouping process, we actually end up with regions that correspond quite nicely to individual proteins, actually. So in a lot of cases, this tends to happen, not in every case. In some case, the regions might be joined so as to join regions from different proteins. And but generally, what this process does is it will as you're smoothing the map, the more globular things will tend to be grouped together. So because this protein is or in this complex, each protein is somewhat globular. It has these two parts, but as the smoothing goes, you can think of a peak being assigned to each protein. So it works well in this case. This has actually been, so again, this is work that I've done as I was entering the cryo-EM field, and it's a part of Chimera and Chimera X. So and I believe it's used to some degree, and basically in the plugin you can create these segmentations. You can select different segments and also group and regroup manually. So this can be pretty useful for visualizing your map and basically starting to interpret what are the different components inside it. Now, just to look at this segmentation process at different resolutions. So there's a lot of maps that even at low resolutions that I think show a lot of interesting details. In this case, if we take this map and we apply the watershed segmentation process, and there again in this case there was no grouping done in this case, we can see that the segments are actually so this is a map of beta galactosidase, and in complex with some antibodies. So it always helps to know a little bit about what you're looking at, of course. But basically, if we look at the segments that are created, some of these segments actually correspond to individual proteins. So these ones on the top here are actually the antibody. But the other segments basically correspond to the beta galactosidase proteins. And there's if you know that protein, there's 4 of them arranged symmetrically. But as we, so let's look at something at slightly higher resolution, 5.5 angstroms. So this is also, I thought, a rather interesting map. This is apoferritin with a bound receptor. But here to zoom in a little bit, and again, these are the watershed regions that are created. What we see is that the segments are now smaller. So here they were larger, low resolution maps. So the signal is smoother, the peaks are wider basically, so you get larger regions. At higher resolutions, you start to get smaller regions. And now the regions start to correspond to small segments of these alpha helices that we can start to see in the map. So when you see these long tubes in a cryo-EM map, they tend to be alpha helices. And here's another map of beta galactosidase at 3.2 angstrom resolution. And now these watershed regions start to, again, be even smaller. And actually, you can see that they start to correspond to side chains or groups of atoms in the backbone. So in maps like this, you can see that some side chains, like this tyrosine, tend to be resolved at this contour level, whereas there might be some other more dynamic side chains, like this arginine, which actually has no segment there. Now, again, this is also based on, there's still this notion of a threshold. When we visualize that, we still that threshold still comes in. So if I lower the threshold, maybe I'll start to see something. But generally, if you lower it too much, you start to see a lot of noise. So you start to see some of the smaller blobs, for example, here. And finally, some of the more impressive advances of this year have been these maps of apoferritin at up to 1.15 angstrom resolution. And now, if we look at the segments, these watershed regions, now the segments actually correspond to individual atoms. So that means that there's actually a peak for each atom and a valley in between two atoms. So that in a way can be used to say that, yes, we are resolving individual atoms in this case. So I thought to show, this range of resolutions. As you might all know, cryo-EM field used to be called blobology in the early days. And I still like to think of it as blobology, but now the blobs are smaller and correspond to atoms. Now, to get a little bit into what do the density values in a map look like around these around actual atoms. So as we got to these high resolution maps, we started to wonder, well, what does the map look like around these atoms? And now this is going to be a slightly different concept than that idea of segmenting, but it's somewhat related. So this segmentation, looking at these peaks and valleys, basically tells us is it resolved or not. This question is meant to address how resolved are they, or basically how fast, or what is the characteristic of the density around each atom, and that can tell us a little bit about how well it's resolved. So one way to measure this is, let's say we start at the center of each atom and we calculate the average map value, and then we start to plant these points around the atom at increasing radial values. So here it's these orange points are at about 0.5 angstroms away from the center of the atom, and they grow outwards towards 1 angstrom, 1.5, and then 2.0. And another thing you'll notice is that here these points are such that they're closer to this atom than to any other atom. So in this case, as you go further away, the points on the sphere with that radial with that radius are only taken if they're closer to that atom that we're interested in than to the other atoms around it. And this is a way to try to isolate out each atom's effect. Of course, it's not doing that completely, because the density values at these points might be influenced by this atom as well. But again, this is still an attempt to try to isolate out the effect on the density of that particular atom. So interestingly, maybe, and it shouldn't be too surprising, with these radial distance plots, or not radial average density, if you will, which another way of calling it is atomic map profile. What they look like is they drop off. So they start off being high at the center of the atom, and they have this drop off. Now, and it also should not be too surprising that they have a Gaussian form. So when we think of atomic scattering factors, let's say we think of them as having Gaussian drop-offs. Usually, more accurate scattering factors tend to consider a combination of Gaussians rather than a single Gaussian. But in this case, if we take a Gaussian function, which is which has this form, and we fit it to this curve, it actually fits pretty well. So the Gaussian function that's fitted to this curve is this black dotted line. So it fits pretty well up to maybe, let's say, 1.52 angstroms, and then here it starts to trail off. It starts to go up again, I guess, possibly because the effect of either atoms or solvent and things like that. Now, interestingly, so let's, okay, so, right, so we parameterize this Gaussian. So the Gaussian has a height, a B, which is the, let's say, the Y offset, and then it has a sigma, which basically represents the width of the Gaussian, let's say. So what happens at low resolution? So this is the same residue and basically a similar map. So these are all maps of vapor ferritin, actually. But this map is at a lower resolution, so 2.3 angstrom resolution. And in this case, if we look at this atomic map profile, we see that, well, first of all, it has a higher height, and that's not too surprising sometimes because maps have different values, different ranges of density values. But more importantly, it has a wider sigma. So, and again, this is not too unexpected in a way, right? Because as we go to low resolutions, we expect the density around each atom to be more diffuse. So they're represented as larger Gaussian. I guess perhaps what was more surprising is the fact that they fitted to Gaussian so well. As I was doing this, I was expecting it. I wasn't sure what to expect, but and again, thinking back to how scattering factors are used to represent atoms and density maps, I thought they would be more like a combination of Gaussians, but it turns out that they're actually very Gaussian-like. Now, to look at some of the more recent maps, this is actually looking at this 1.22 angstrom map of also vapor ferritin. And again, here we see that the atoms are actually separable at a certain contour level. Whereas if I were to look at the separability in this map, they might not be separable at a certain contour level. But in this case, they are. But if we look again at just the density values around it, they still fit very much of, they still have very much of a Gaussian like appearance. Although here it starts to trail off a little bit sooner. So the tails of these are a little bit mysterious still, if you, I suppose. And again, this might be where the influence of other atoms start to come into play. But for the most part, initially, they still have this Gaussian-like shape, and you can fit Gaussians to them with pretty low errors. So if you were to take the errors between the atomic map profiles and Gaussians, you tend to get an error of maybe 2-3%, let's say, up to this point where they start to diverge. So okay, so as we were doing this, as we were looking at this, and I remember presenting this in a group meeting at one point, and basically I was suggesting that, okay, so we can sort of assess the resolvability of atoms in a map just by fitting Gaussians and calculating the sigma from the fitted Gaussian. And then you have a nice way to sort of, in a way, back calculate, or in a way you're trying to, you're fitting Gaussians, you're trying to sort of figure out what is the resolution of the map just from these curves. And then somebody asked the question, well, what if the atomic profiles are not Gaussian? So it was a very good question, and it got us thinking about, okay, what if, so right, so if you're trying to fit a Gaussian to a curve that doesn't look Gaussian, you know that you tend to get random results or unexpected results. So that wouldn't work too well in those cases. And not all atoms, as I'll show a little bit as we go on, not all atoms have these nice, they only have these nice drop-offs or these Gaussian-like profiles if they're well resolved. And if it's not well resolved, then it's unexpected, really, what you get. But this got us to the idea of a Q-score. Okay, how do we quantitate, or how do we take these atomic map profiles and sort of give a value as to how close are they to a ideal Gaussian, let's say, to what we would expect to see if the atom was well resolved. So, so one way that turned out to work well for calculating the score is to take basically take the atomic profile, take the map values around an atom at increasing radial distances, and correlate them to a reference Gaussian. So the reference Gaussian is what you would expect to see if the atom was well resolved. So so basically the Q-score is calculated for each atom in this way. So again, we build these balls of points at increasing radial distances, and we interpolate values from the map at those points and correlate them to values for a reference Gaussian. And the reference Gaussian has to be given, has these three parameters. So the, again, the height, the y offset, and the sigma. And the question was, so the height and the offset basically are taken from the map value. So we look at all the values in the map and we see, okay, what's the highest value, what's the lowest value, and basically the height is taken as let's say the mean value plus some amount of standard deviations of the value. So that's to if you take the highest value in the map and use that, there might be some noise spikes, so that's not always great. So so the height is set as 10 standard deviations above the mean, and the offset is set as 1 standard deviation below the mean. So that's because if you go too far below the mean values, then you start to really just hit noise. So now the sigma is the more important parameter. So because as you have lower sigmas or lower widths, then that means that this reference Gaussian gets sharper and sharper. So originally the sigma was actually was set to 0.6, but what tends to happen is if you're correlating a wider peak to a sharper peak, then you get a lower score. So we have to adjust it basically to the highest resolution maps that we have. So sigma of 0.4 basically looks a lot like these atomic map profiles that we see in these highest resolution maps that we have to date. So so if we take these so if we take the atomic, or if we take the values around the atom in this map and correlate them to reference Gaussian, in this map we get a Q-score of 0.97. So the highest value, because this is a normalized cross correlation, the highest value you can get is 1. So we get pretty close to the highest value in this map. And in a low resolution map, so this is a 1.34 angstrom map, now the map values around the atoms start to drop off as the drop off is a little bit wider. So now we start to get a lower score. And as we go to lower resolutions, we get even wider profile, so wider drop off. So then the score keeps going down. And these so as you go to lower resolutions, you will tend to get a lower score. And I don't have I didn't show it here, but let's say the map values were to be pretty flat or sort of even increased, then you would get a very low score, so close to 0, up to a lowest of -1 possibly, if let's say the it was actually a reverse Gaussian, if there is such a thing. So so here are some more examples. It's actually similar, yeah, so similar examples. So now going to even slightly lower resolution of 3.1. So in this case, we can see that some atoms are even seem to be outside of the contour. So and one thing to note here is that the score is not really dependent on any contour. So there's no threshold level that's technically used in this calculation. So even if this atom is actually outside of the contour at this threshold level, again, that doesn't affect it. So so the Q score is lower because at this point, the density is dropping off either more slowly, or it's even flattening. So then we get lower Q score. So this can give us a rough indication of basically how well atoms are resolved. Now, another thing we did was to take a number of maps and models from the EMDB and basically calculate Q-scores for all of the atoms in the model, and then plot the average Q-score as a function of the reported resolution. So this is the way of measuring resolution I talked about at the beginning, where we look at two independent reconstructions, and generally that right. So that measure, the report resolution, is usually a pretty good indicator of the level of detail that you would see in the map. And we can see that the average Q-scores actually, on average, correlate very well to that resolution. So this told us that Q-scores are actually could be a pretty meaningful way to actually evaluate the resolvability of different atomic features, with the added advantage that you can apply it to individual atoms a bit more easily. So with the estimated resolution of a map, that corresponds to the entire map rather than to smaller parts. So we can go into calculate the actual resolution in smaller parts by breaking the map up and doing something similar, looking in Fourier space and signal versus noise. But that's a little bit more tricky, and especially it's hard to do it for smaller and smaller features, because you have to look at a certain number of points around an area to be able to estimate the resolution using that approach. Whereas with the Q-scores, we can actually, so again, we can do it basically for each atom. One thing we see that's interesting is, so we can calculate Q-scores for side chains, or atoms or backbone atoms, or for the entire residue. So one thing that's somewhat interesting is that the backbone Q-scores drop off a little bit more slowly. So at this low resolution, the Q-scores for the backbone atoms is higher than the side chains, whereas at these very high resolutions, the side chains are just as well resolved. And this is something that we also see, that's a usual trend, that as we get to lower resolutions, usually the side chains start to disappear first, and the backbone tends to be a bit more visible. We can also apply this to RNA. So it doesn't really matter what type of molecule the atom comes from. So I think the highest resolution RNA model to date is 1.9 angstroms. And this comes from a protein RNA complex. There's actually only 3 RNA bases in that map, but we can still use it to calculate Q-scores, and basically we see the same trend. So we also see that the Q-scores tend to drop as resolution decreases. So here's so again, here is that trend. So here we have each dot corresponding to a map and a model in the EMDB, and basically what we can get from the Q-scores, let's say something a map like this, with the high Q-scores, we can expect to see well-resolved side chains and well-resolved atoms in the side chains and backbone. And then as we get to lower Q-scores, we start to see side chains a little bit less resolved, as well as this helix has just a bit of a pitch here that we can see, or the grooves. And then something with a low Q-score is where we start to see just more smooth features, and not as much side chains and so on. So what's interesting, I think, about the Q-score is that you can look at some of these outliers and wonder why does this model and map have such a low Q-score that's not close to the expected value. So if we look at one of the well-resolved helices in the map, in that case, we can see that that it has you can see some of the side chains, you can see a bit of helix pitch. So it's kind of curious that it has such a low Q-score for it that doesn't fit that trend. So the Q-score should be closer to 0.55 in this case. So when I opened that map and the model, what I noticed is that the part of the protein is well resolved. So here we can color the backbone or the ribbon using the Q-score. So the residues with higher Q scores are green, and the residues with lower Q scores are red. And we can see that some parts are not resolved at all, whereas some parts are resolved. So this tends to, and this is not too uncommon. A lot of maps tend to have some variability in some regions in which residues might not be as well resolved in others. And a lot of the times the resolution estimate can use some masking. So they might have masked only this part of the map, let's say. And so this part of the map actually has a Q score that's more in line with what the expected Q score for that resolution is. Whereas another part of the map and model is not so well resolved. So in that case, those Q scores are much lower. So we end up with this average value in between. So this can be a useful way to visualize what are the Q scores throughout the models, what are the different resolvabilities, and yeah, and it can sort of flag some of these maps and models which maybe are resolved better in some parts and not so well in others. So here's another example of applying Q scores per residue Q scores to a map and a model. So what we started to do, and we think it's pretty useful, is to plot these Q scores as a function of residue. And you look at these plots, and we also plot, okay, what is the expected Q score for a certain resolution? And that's this gray bar that we see here. And basically, wherever the Q score is higher than this expected Q value, that means that it's as you would expect. But then you might see these dips, which you can sort of look at and see what's going on there. So in this case, this dip corresponds to this part here, which is colored in red. And if we look at the map and the model there, we can see that the map is indeed not too well resolved in that area. So we can, and if the map is not too well resolved, then there's not too much you can do, but just know that that part of the model is maybe not so certain. But in some cases, so here's a case that we started to look at. This is a map that's related to the coronavirus. And in this map, when we plot the Q scores versus residue, we saw this big dip here. So I started to wonder, what is this dip from? So if you look at the model, there's actually some density there. So it corresponds to this loop, which once you look at it, you can sort of see that it doesn't fit so well into the density. So out of curiosity, I was wondering if I could fit this better. So I ran the model through Phoenix Refine. And it improved the Q scores a little bit, but still not too well. And then I also tried to go in with more of a manual intervention, where with this Zold and Chimera X. And here you can do a molecular dynamic simulation and start to pull on atoms and try to fit them better into the map. But I have to say, I kind of failed in this case. I couldn't actually get it to fit any better. This is maybe when I gave up, this is probably the best that I could do, and improve the Q scores a little bit. But so I guess what I'm trying to show with this example is that just because a Q score is low, it's either that it could be two reasons. One, that the map is not resolved, or maybe the model needs to be fitted better. So if the model needs to be fitted better, that's sort of that's another challenge, so to speak. I think it's still a bit of an open problem why possibly how to better fit models in cases like this. Because I guess the protein backbone has a lot of flexibility, but at the same time, it still has to fit certain dihedrals, angles, and things like that. So there's still some limitations in trying to fit it well to the map. But again, that's sort of a really interesting active area, I think, to how to refine and how to optimally build models in some cases like this. Okay, now to look a little bit at ligands, and this is an example that Kathy Lawson brought up as we're starting to prepare for the next EMDR project challenge. So these are two maps of beta galactosidase. The top one is at 2.3 angstrom resolution. So we can look at these ligands that are in these binding sites, and this is the density around the ligand that's masked out. and in this case, the average Q scores for this ligand is 0.73. And I thought it was interesting that in this map, even though it's higher resolution, the Q score is actually a little bit lower. And visually, you can see that the density is not quite as well resolved. And again, that's perhaps it's not too uncommon because, again, when we give one resolution number for the entire map, that doesn't necessarily represent the entire, doesn't represent every part of the map, it just represents the entire map. In this case, for some reason, these ligands are not as well resolved as in this case. So there's probably a lot of interesting experimental factors as to why this happens. But again, sort of the take-home message is I thought it was kind of interesting that the Q scores can actually catch such, give you a score that better reflects what is the resolvability of each atom at different parts in the map. Okay, now to get a little bit into ions and water. So as I mentioned, I would talk about this a little bit. And recently we started to get interested with these high resolution maps. We started to get interested in, are we really seeing waters and ions? Because if we go back and look at these blobs or these watershed segments around a model that was built into the map, we start to see that there's a lot of these small blobs that could potentially be ions and water. So one way to model the waters, we can go back and take, well, what we did initially was to take X-ray models that were fitted to the map and look at where the waters and ions were placed in the X-ray model, and then look at, are there peaks there or not? So that was the initial step. The second step was to actually try to build these in somehow manually or automatically. So we came up with this procedure, which we called SWIM, for segment-based water and ion modeling. And basically the procedure is to take these blobs, these peaks, and we look at a threshold level of 2 sigma about the background. So that means that you've excluded, hopefully, a lot of the noise. And looking at each blobs, first of all, ignore it if it's already an atom that's in the model. Ignore it if it's too close. If it's too close to, say, a carbon or any other atom, like let's say if it's less than 1.9 angstroms away, then it's ignored. And also we can use Q scores here to pick out the blobs that are actually more, basically look more like Gaussians, because you might get some funny peaks that are either too sharp or kind of flat, and those are probably less likely to be waters, or what you expect to be an individual water or ion atom. And then basically look at, okay, what are the distances between those remaining blobs and polar or charged atoms, so oxygen or nitrogen atoms, because the ions and water tend to have more stable positions closer to, closer to such atoms that are polar. So in particular, we took some criteria that were that was actually published recently in a paper. It was called the UNDAUs, or basically because it would take any deposited model, and it would say that it would apply some criteria saying that if it's too close to such a polar atom, it's more likely to be an ion, and if it's just the right distance away, let's say more of a hydrogen bond distance away of 2.8 plus or minus some value, then it's more likely to be a water. So this is by no means a very thorough analysis yet of what the difference between ions and water is, but it's sort of a start. And here are some results of applying this procedure. So first, starting with this high resolution map of 1.15 angstroms. In this case, this is just looking at these 3 small area around these 3 residues. And having looked at the crystal structure before, this is apoferritin again. So having looked at the crystal structure, this was identified as a ion binding site in particular. So in the crystal structure, there was a magnesium ion placed there. Now in the cryo-EM maps, we're not too sure if magnesium is actually in the buffer. So we're not too sure if this is really a magnesium atom ion. It could be zinc. It could even be iron, right? Because apoferritin binds iron ions. So it could potentially also be that. So there's still a bit of some questions related to what type of ion is it, because these ions tend to have similar properties. So they could bind in similar ways to in such binding sites. But so anyway, so the procedure finds that this blob in the center is likely an ion, and that this actually corresponds well to what was in the crystal structure. And that's because this was within, let's say, maybe 2.2 angstroms away from these charged oxygens and this nitrogen. And then some other blobs around it placed them as waters. So that's because, again, they were probably more within something, a range like 2.8 angstroms away from such polar or charged atoms, which tends to be more water. Now, the interesting thing that also came out of this is that if we create these plots of basically, these are radial distance plots of the number of atoms found at a certain distance away from certain types of atoms. So the red curve shows the number of, so this was done for the entire protein, the entire apoferritin protein. So there's about 100 and let's say 120 water atoms. So these are, the red is the water atoms that were within 2.8 angstroms away of an oxygen atom in the protein. So it's interesting that you get such a sharp peak because, again, it could have placed the water at anywhere between 2.4 and 3.3, but really it found blobs that were predominantly at about 2.8 angstroms away. So it's kind of interesting that you get this peak rather than something that's more flat in this case. So that means that a lot of the waters that were placed were actually at that, if you will, sort of equilibrium or optimal distance for waters away from a polar or charged oxygen atom. Now, the green curve is basically the distance, is the number of ions that were placed and at certain distances away from also oxygen atoms. And in this case, now the dotted lines, you can there's a lot fewer ions that were found. So the dotted lines fall on this axis. So about five ions were placed at a distance of about 2.4 angstroms away. And again, not too many atoms. Let's say one ion was placed at 1 angstrom away and 0 were placed at 1.8. So not too many blobs were found that were too close. So it's interesting to see these peaks for ions and waters. And again, and that sort of speaks to the sort of the chemical properties of the distances you would expect ions and waters to be away. So in a way, this placement procedure produces that in a way somewhat well at this resolution. Now, something at lower resolution, it still found ion in the similar position. Now, the waters seem to be have slightly different positions, but in this case, the peak for the water atoms is a little bit smaller. So it only placed about 80 water atoms at about 2.8 angstroms away. And it placed 50 something at 2.6 angstroms away. So here the distances between the waters and the oxygen atoms in the protein tend to have a bit more variability, but there's still not too many that were placed at 2.4. So as you would expect, because they're too close in that case. So it didn't find too many blobs, if you will, at those distances away. So again, that kind of tells us that this procedure is actually finding something that's chemically sort of relevant. It's something at 1.5 angstroms, 1.75 angstroms away. Again, the peaks start to get a little bit more diffuse. So as you get to lower resolution maps, this tends to happen is that features tend to be a bit more diffuse and you can't place them with quite as high accuracy. So So we see the peak for the waters go down and become more diffuse. But interestingly, the peak for the ions, again, it didn't place too many ions in this case, maybe 5. And a lot of them did correlate with the ion positions that were placed in the crystal structure as well. So there was some, okay, yeah, and here is this is looking at the crystal structure and the 2FO minus FC map. And in this case, we can see that there are some ions and waters placed in that map as well. And again, the ion position correlated well with what we saw in the cryo-EM maps. And although interestingly, you see the water positions are still kind of a bit more variable. So water might be a little bit more dynamic. It might depend a bit more on the actual conformation of the protein. But this was sort of in a way, kind of the more reference, if you will, where we see that in this crystal map, because things that are likely at a more sort of lower energy state, more equilibrium state, we see a very sharp peak for waters to oxygen atoms at 2.8 angstroms, and also a sharp peak for ions to oxygen atoms at 2.2. Now, I didn't talk about the other curves. There are these water to nitrogen atoms and ion to nitrogen atoms. And we do tend to see that there are some ions and waters close to nitrogen, but definitely not as much as to oxygens. So, and I think this is probably somewhat known that water will tend to sort of favor hydrogen bonding to oxygens rather than to nitrogens for some reason. But yeah, you can see that there's not too many waters or ions that are placed close to nitrogens, and that's because again there are no blobs or there are no peaks around the nitrogen atoms, not because right. So this procedure basically looks at the peaks and the blobs around each atom, and only places something if it's within a certain distance away. So and it ignores small regions that are noise. So again, but I think because the plots are quite similar to what we saw in the X-ray map, and in a way it sort of starts to show that indeed what we're seeing in these higher resolution cryo maps correlates well to water and ion positions. Okay, now the last thing I'll talk about, and this is something even sort of more new that we started to work on, is basically looking at are Q scores and B factors related? So one of the responses we got when we published the paper from a reviewer was, aren't Q scores just like B factors in a way? Because Q scores really measure how diffuse is the density around an atom, and B factors in a way do the same thing. So if you had a map and a model that looks like this, some atoms again are not too well resolved, so they're in lower density, so you don't see a contour around them at this threshold level. So for atoms like this, you tend to assign higher B factors because the reason that they're not so well resolved is they might just be moving around a little bit too much. So if they're in a different position in each particle in cryo-EM, that density will be a bit more smeared out. There could be other reasons, like some of these residues might actually be suffering from radiation damage. But ignoring things like that, you can make that, sometimes you can work with that assumption that the more dynamic an atom might be, the less resolved it tends to be. So, and I think B factors are quite useful in crystallography, and we're starting to sort of look at how can we apply them to cryo-EM as well. And basically one way to apply them is to see what the effect of a B factor is if we take a model and we calculate some B factor for it, we can use those B factors when we generate a model map. So this contour here, this surface shows a model map or a map calculated from the model using Phoenix F-map. So with Phoenix F-map, it takes the position of each atom and it applies some scattering factors, and then it also applies a B factor. So if you have a calculated B factor for that atom, it will apply that. And then things that have higher B factors will tend to be more spread out, not as sharp. And then it will apply a resolution factor as well. So which is usually in the form of a Gaussian blur. So the lower the resolution, the more you blur that map, and you get something that's smoother. So when I tried Phoenix F, I started in this example, I tried to calculate B factors using Phoenix Refine. So when you run Phoenix Refine of a model into a map, it will try to calculate these B factors or ADPs, as they're called in some cases, or atomic displacement parameters. And then you can plug those back in and calculate the model map. So in this case, though, it seemed like the B factors still didn't quite account too well for the fact that this residue or this side chain is really not too resolved in the map. So calculating the B factor from the map in this case was such that it's still reflecting that this residue should be probably maybe more resolved than it should be. And there's probably different factors for that. I think when you calculate B factors, you tend to use constraints. So usually you don't have too much information about each position in space. So you might want to constrain it to say that the B factors of this residue should be similar to the B factors of this residue, because how much residues are moving tends to be influenced by how the residues are connected together. They tend to move in similar ways, either be more mobile or less mobile. So there are these constraints that are applied, which in this case made it look as if this residue maybe is resolved better than it should be. In this case, I also tried RefMac. So interestingly, with RefMac, it calculated B factors in such a way that the model map was definitely more smeared out in this case. So at this contour level, you don't see too much of this aspartate residue. But there's still a couple of things that were a little bit off. So for example, this nitrogen atom here appears to be resolved, whereas in the cryo-EM map, you can see that it's not, it's in a lower density for some reason. It's not as well resolved. And also this glutamine, the nitrogen atom seems to be not too well resolved. It's kind of sticking outside of the contour level at this point. So being encouraged to try to relate Q scores to B factors, this is something that it's sort of a kind of a first attempt at relating Q scores to B factors. So obviously Q scores are range from, again, from -1 to 1, with 1 being well resolved and -1 being not well resolved. So these have to be turned into the scale space of B factors in a way which is actually quite the opposite. So the actual formula for this was F times 1 minus Q score. So that means that if the Q score is 1, this will be 0 and you get a small B factor. And if close to 0, let's say, whereas if the Q score is low, then you get something closer to this F value. So this F value is pretty much empirically determined at this point, where we tried different values and we saw what worked better. But in this case, if we apply these B factors, now we can see that we kind of see what the cryo-EM map was showing, is that this aspartate is not resolved. So this is all smeared out. And then this nitrogen atom is not resolved, whereas the oxygen is a little bit better resolved. So as we see here in this case, the oxygen seems to be in a higher density. And actually the modeling here is also sort of in question to some degree. This could sometimes be flipped. So this might not have been modeled, it might not reflect the exact conformation, let's say. And also it still gets some things wrong. So because the model maybe doesn't go exactly through the right density, here you can see that with Q scores, like this bar seems to be less resolved. So it's not perfect, or it's not necessarily better in every way, but this is just another way of basically trying to address this issue. Is how do we annotate our model? So when you download a model from the PDB, if you don't look at the map that it came from, it might be easy to assume that the model is perfect in every in every atom that is placed, it's at exactly that location, which is obviously not the case. So, so I think we're starting to realize that it's important to start to try to annotate these models, much like crystal structures were annotated with B factors. We might want to start to annotate them with something like a B factor as well for cryo-EM. So this might be one way to that we're still exploring how to do it and so on. Okay, so so that's so that's it. So to look back at what we talked about, we looked at basically this idea of segmentation and these blobs and how they can be used to decide in a way. It's more of a qualitative, really sort of look at how things are resolved in a map or not. And I think it's a cool way, if I can say so, because I've been working on it. So it's a cool way to look at your map and start to pull out the different parts that are visible inside it. And then we looked at Q-scores as this per atom quantitative measure. And we saw how we can use it to look at resolvability of different features within the map, applied it to different scenarios, proteins, RNA ligands. And I think in terms of the future, basically, it's something I've been thinking about how to do, and it's basically, can we actually apply this to more maps and models, even possibly the entire EMDB? And I think this can start to tell us a bit more about how do the models that were deposited, how do they represent the actual cryo-EM data, and not as a single number, but overall, like throughout the model. I think it's starting to become apparent that we can use this possibly as a search. So if you're searching for a ligand, for example, for ligands, you might want to know how how well resolved they are, or how well does how likely is the ligand to be in a good sort of bound state, and this can help with things like drug design and things like that. And in this area of annotating models with B factors or ADPs, or in relation to Q-score, I think could also be interesting for the future as well. So I'll finish with acknowledgements. So so I've had the great pleasure of working with Chu's lab for a while now, and these are some of the current members that are very great to talk to and have group meetings with and so on. And of course, as Kathy has mentioned, we've been collaborating as well on this EMDR project. And they've had a lot of influence as well about on this work as well. So I'm very thankful of that. And thank you for listening. And I think I was expecting to finish a little bit earlier, but I guess I did go a little bit longer than I thought. I don't know if we want to do some questions now, or
I think we probably have time for maybe one question, if there's any intrepid person in the audience.
There's something in the chat.
Oh, okay. Well, unmute and go ahead and ask it.
Hi, my name is Robert Young. I do a lot of work with computational modeling for circular DNA, and I'd like to expand to a more sequence-dependent role. I've seen some work that was done in the past where you could take cryo-EM or cryo-TM images of supercoiled DNA, but you lose the ability to try to ascertain sequence tracks. It is really difficult. I understand it is difficult to find sequence tracks with cryo-TM or cryo-EM images. This Q-score work that you have, is there a way that you could maybe use knowledge-based potentials of what we know about specific configurations of dimer, or maybe even like tetramer step information, and apply that to maybe looking at ways we can accurately locate with a cryo-EM set of data what tracks of DNA we're looking at? If that's something that's a little too large scale, or we lose that, it's not as nicely structured as like a series of protein residues. But is there a way that we can utilize Q-scores for DNA, better DNA knowledge information? Sorry.
Yeah, I think so. Yeah, with DNA is interesting, and looking at Q-scores in DNA, what we noticed is that, again, we see different properties for bases and backbone of the DNA. So sometimes the backbone tends to be more flexible, whereas the base pairs are a bit more resolved. So I think it speaks a bit to the different dynamics of different molecules. Yeah, I'm not sure about tying knowledge based in this. I think this was even more simplistic than doing something knowledge based, where we really just looked at the raw density values rather than trying to say this, it looks like this in certain ways. I suppose the only knowledge that's really built into this is that idea of the reference Gaussian, that atoms or even groups of atoms tend to have these Gaussian-like drop-offs, and basically it reflects that sort of property. But yeah, yeah, I'm not sure if that's.
Greg, let me pose to you a question from the chat from Shenghua Xiao. What about alternate conformers? So you could have a case in which the basic assumptions in the Q score would be violated because you have you could have something that's very highly resolved, but the peak height is lower because of low occupancy, and you could have two peaks that are very close to each other because they're not co-occurring in the same copy of a moiety. So what would be the way to proceed in the case of alternate conformations of a side chain, for instance?
Yeah, that's a great question. I went back to this slide here because I remember seeing an alternate conformer in this case. So you can see this arginine is kind of goes off in two different directions here. So yeah, indeed, in those areas, you start to see that the density looks a bit more maybe noisy, very peaky, like sharp peaks. So in that case, I guess it doesn't do anything too intelligent. It really just looks at the position of every atom and says, okay, in this conformer correlates better with the peaks, and the peaks look more atom-like than in this conformer. So you can use that to kind of pick which conformer is more resolved than the other. But yeah, I think it's an interesting question in that I'm not sure it addresses too well that there might be. So in this case, for example, there is a part of the side chain in both conformers where the atom position is the same. So in that case, you would expect that density, because it represents more particles, is still well resolved, is still atom-like and not as smeared out. Whereas in the other parts where the atoms are different in each particle, they start to, you see that they're not as well resolved. So you will get a lower Q score in those areas. But yeah, that's something to think about. A very interesting question. Yeah.
Unfortunately, due to time constraints, we'll have to close it there. I'd like to remind the quantitative biomedicine grad students to hop on the Zoom call for the post-seminar discussion with Greg, which will be a little bit abbreviated due to his scheduled meeting with speakers today. But thanks everyone for attending. I'm sorry we didn't get to more questions. And Greg, we really appreciate having you here virtually. Hopefully we get you over the border sometime in the future to come and visit us physically. So as always, thank you very much.
Thank you.